The last thing we need to talk about in the brainstem is something called the reticular formation. The reticular formation is a net-like grouping of neurons that appears to be really concentrated throughout the brainstem. Not just the midbrain, not just the pons, through that whole section. We refer to this as, again, the reticular formation. The word reticular refers to net-like, all right? Throughout anatomy and physiology, there's often references to something that has a reticular appearance, and that means it has a net-like appearance, like a fishing net, okay, or fishnet stockings, right? The purpose of the reticular formation is to receive incoming information, right? It's receiving afferent axons, afferent incoming information that deals with arousing and maintaining consciousness, so keeping you awake. So, when your reticular formation is in fact receiving this information, it's keeping you conscious, it's keeping you awake, giving you the ability perhaps to focus. It receives and integrates input from all parts of the brain and the spinal cord. So, if you're having difficulty sleeping, it makes sense because you're getting all of this incoming sensory information. I'm too hot, I'm too cold, my shoulder hurts, the sheets are uncomfortable, whatever it is. It's all coming through all of these different various sections of the brain and spinal cord and sending this afferent, this sensory information in through the reticular formation. Next up is the diencephalon. Diencephalon is the second of our four regions of the brain, right? We had the brainstem, diencephalon, cerebellum, and the cerebrum. Contained within the diencephalon is something called the thalamus. It's an oval structure above the midbrain. So again, think about that sagittal section diagram that we saw, and the thalamus is going to be above that, superior to that. It consists mostly of gray matter. Gray matter is going to be unmyelinated fibers, right? Either cell bodies or unmyelinated axons and dendrites that are organized into nuclei, clumps, with two larger lateral portions connected by the intermediate mass. The best way I can describe this to you is as a yo-yo. Okay, the two larger lateral portions are the yo and the yo, right? The two main parts of the yo-yo. And the intermediate mass is the stick that connects the two parts of the yo-yo that we put the string around, right? So that's what this is going to look like. It is a major sensory relay station. And again, this makes sense. It's in that pathway from the spinal cord up through to the higher, quote unquote, higher levels of the brain, the cerebellum and the cerebrum. It influences mood. This is the first time we speak of this, but understand it's the first of many. And again, as humans, we think our mood is controlled and moderated by the cerebrum, by our upper thinking ability. This isn't necessarily the case. The thalamus is the first of non-cerebral areas that are going to have some influence over our mood. So we've got three graphics here. The top left, it says thalamus. This is a mid-sagittal section. We're cutting it right down the middle. So what we're looking at is the flat portion of the yo-yo, right, of one of the halves of the yo-yo. If we look at the other diagram labeled thalamus, that's showing you the two yo-yos, right? And the mid-sagittal section, I'm cutting it right down the center, so I'm looking at the flat portion of the yo-yo. And the next is the hypothalamus. And again, the thalamus, the right graphic, and the hypothalamus are both what we call coronal sections. We're kind of slicing it like a loaf of bread. And that's showing us the hypothalamus, which is another section of our diencephalon. It lies below the thalamus, and that's exactly what hypothalamus means, and above the pituitary gland. The pituitary gland is kind of stuck on a stalk below it, okay? It has many, many functions related to homeostasis. That whole process of keeping our body within that normal range that we talked about way, way back in January. One of the ways it does that is to control and integrate ANS activities, the autonomic nervous system. The autonomic nervous system, we'll remember, is made up of the sympathetic and the parasympathetic divisions. So this is kind of monitoring, are we amping things up? Are we getting ready for that fight or flight, or are we backing them down, doing that rest and digest? It controls normal body temperature, right? So if you're running, if you are in a refrigerator, right, you're in a cold area, your hypothalamus is going to kick in and adjust homeostatically what's happening to keep your body within that normal range of temperature. It's associated with feelings of rage and aggression. And again, here we are, not anywhere near the cerebrum, talking about mood, talking about rage, talking about aggression. It regulates food intake, and this is really fascinating to me. It contains something called the hunger center. What this is telling us is there's, this group of neuron cell bodies that will inform your body, make you go, I'm feeling a little hungry. I think I could use a snack. That's when the hunger center turns on. Now, the hunger center is turning on. It's just telling you, okay, it's time to get something to eat. However, there is also something called the satiety center. The satiety center is kind of the opposite to the hunger center. It's telling you that you are in fact satisfied. You've had enough. How many of us, myself included, have ignored that satiety center? We all have. Right? We've all sat and had a wonderful meal, or maybe not such a wonderful meal, all right, simply with a bag of chips or a bag of M&Ms, saying, oh, God, I couldn't possibly eat anymore. All right, maybe just another. Oh, my God, I'm so full, I'm going to get sick. Well, maybe I'll just have one more, right? So as humans, we have the ability to ignore that. However, if we paid attention to what our hypothalamus is telling us, theoretically, no one would be overweight, because we're in tune with what the neurology is telling us. Now, the problem with the satiety center is kind of simple. It takes about 20 minutes for the signal from the stomach, right? And we're going to know it because the stomach expands as we eat. It takes about 20 minutes from when we start eating to when we are in fact full for that signal to get to our hypothalamus to say, okay, enough is enough. That's why oftentimes with dieting and trying to monitor how much we're eating, it's said that it's a good idea to, just before you have dinner, typically a big meal, to have a nice big glass of cold water. That's because it's going to start stretching that stomach, and it's going to jumpstart that signal and get us moving towards that feeling of fullness faster. That's also why your mom, or maybe my mom, would tell us, okay, in between bites, put your fork down. Chew your food 20 times. It's not so much to chew it up to make it small enough to swallow, although that does help. It's also to slow you down. Because if you're just shoving food in your mouth really, really fast, you tend to get over full, because you're outstripping the ability of the satiety center to catch up with how much food we're shoveling into our mouths. We also find a thirst center in the hypothalamus. This is why oftentimes, again, late afternoon, say, oh my God, I'm starving, I'm really, really hungry. Well, studies have shown that we're probably not hungry, but we are in fact dehydrated. So the thing to do is, before you reach for that bag of chips, before you reach for that Snickers bar or whatever it's going to be, take a moment, have a big glass of water, wait a couple of minutes, and then see if you're still hungry. All right, so let's see, is it the hunger center sending me a signal, or is it the thirst center sending me the signal? Next, we have our cerebellum. That's our third section, our third region of the brain, right? We had our brainstem, we had our diencephalon, and now we have our cerebellum. Cerebellum is located behind the medulla oblongata. and the pons. And remember, the medulla oblongata and pons are kind of one on top of the other. And below the occipital lobes of the cerebrum, it's kind of tucked underneath the back end of the cerebrum. It attaches to and communicates with the rest of the central nervous system through three main nerve tracts. So here's a diagram showing us where the cerebellum is. To the left is the anterior view. To the back is the posterior view. The cerebellum is tucked underneath the posterior view. This is the sagittal section, mid-sagittal section of the cerebellum. Now, if you tilt your head to the left, right, you're going to see what is referred to as the arbor vitae, or the tree of life. That's showing you those white sections, that's white matter, forming kind of a tree-like appearance that we do find in the cerebellum. We're going to find gray matter on the outside, white matter on the inside. That's why with that diagram, the white matter is going to be the branches, and the gray matter is on the outside of the structure. The cerebellum is associated with subconscious skeletal movements needed for coordination, balance, and posture. These are those movements that we are unaware of. Right? We talked often about conscious motor control. This is subconscious. This is you're not paying attention to it. This is your ability to stand upright without having to think about making corrections. Oh, I'm tipping over. Let me move over. Remaining upright in a seat and not tipping over. That's where the cerebellum comes into play. When there's damage to the cerebellum, it can result in three main things. The first is ataxia. Ataxia is the loss of body control, right? The loss of the control of body movements. Next is the disturbance of gait. Gait is your ability to walk, right? Your gait is how you step, right? How long your steps are, how quickly you walk. That's gait. Disturbance of gait is your ability to walk properly. And last but not least is severe dizziness. So if someone experiences damage to the cerebellum, their muscular control, their muscular coordination is going to be very, very severely compromised, as will they experience severe dizziness. Which brings us to this. Right? If we look at those quote-unquote symptoms, or the effects of damage to the cerebellum, it sounds very, very, very much like a long night at a bar. And that's why these sobriety tests work, because the cerebellum does in fact, alcohol does in fact affect the cerebellum. And because of that, we can use motor coordination to determine whether or not someone is in fact compromised due to use of alcohol.